Deoxyribonucleic acid, or DNA for short, is the molecule that carries the genetic information of all living organisms. And what I'll be doing today is exposing this DNA in the strawberries and also extracting it. But first, to get to this DNA, which is stored in the nucleus of a cell, we have to pass through a few barriers. What are these barriers? So let's look at the plant cell and try to figure out what needs to be destroyed to get to the DNA in the nucleus. This is a plant cell. Okay. So the first thing that needs to be destroyed is the cell wall. And how we're breaking down the cell wall is by pulverizing the strawberries, which is part of the process of what I'll be showing you. We'll be pulverizing it, which what it'll do is actually expose the strawberry cells and also structurally damage them. And now once it's structurally damaged, we are on to the plasma membrane, which I'll be destroying by using soap. And so soap will attract the phospholipids and also the proteins in the plasma membrane, ultimately severely damaging them and exposing the inner parts of the cell to the environment. So after the inner parts of the cell are exposed, we then move on to the nuclear membrane, which is the closest barrier to the DNA. And so this nuclear membrane is destroyed by sodium chloride, which is also salt. So the sodium ions will break down the protein and then destroy this nuclear membrane and expose the DNA to the environment for me to extract by using alcohol. You're now probably wondering, how did I come about this idea in the first place? Well, I was walking about my garden when I suddenly came about a bunch of wild strawberries. So I took them in, put them in a Ziploc bag, and started pulverizing them, which is also the first step of exposing the DNA in the plant cells. This is where we're breaking down the cell wall. And here's the finished product. As you can see, it's all gooey and yucky now because it's all crushed. Next, we're preparing a soapy salt solution that will be destroying the cell membrane and also the nuclear membrane. We'll then mix it all up and put it into the Ziploc bag that has the strawberries. Okay. Now we're mixing it around again, and this is actually the most important part of this experiment, because this is where we're getting rid of the cell membrane and also the nuclear membrane. Next, we're straining out all that excess gunk to get the DNA. So I strained it out about 3 times to get this. Now I take my ice cold isopropyl alcohol and pour it into the cup. I stir, 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 and out comes the DNA. Ta-da! Looks quite yucky, right? But I'm here holding the genetic code of life in my hands. Yep, with that our experiment's finished. Thanks for watching, and bye bye.